is going to be about actually making a blot, but there are two important things I forgot to say about running the gel and sample prep. One of them is in your list of things that you add into the sample. So there was dye, there was reducing agent, there was detergent. There is also something that is a density increaser. And usually this is something that is very dense itself and sinks down in water. Normally this will be something like propylene glycol. And the reason that you put that in there is that when you're loading samples, you're going to have a pipette tip carefully held right there that is not touching the sides. You're just holding it there. And you're going to push your thumb so slow, you hope you are, you're going to push your thumb so slow, and your sample, because it's got a density increaser in it, will fall down to the edge of the hole, the well. It'll fall down to the bottom of the well, and then as you keep pushing really, really gently, it will fill up. And it will not all disperse out here. You need the density increaser, which is usually propylene glycol, to get it to fall into the well, and you need to not oops your thumb and squirt it really violently, because then it will go everywhere. The other thing I want you to know is that in a realistic gel, in a real research lab, the place where the dye, the place where you actually see the dye migrating, because realistically that's what you see, not bands like this normally. The place where you see the dye and you check how far it's gone is called the dye front. Like storm front, it's a dye front. And when the dye front gets down toward the bottom, you usually know that you can turn off the gel and you have separated your proteins far enough. So that information is pretty important. It's really important actually in practice of preparing protein gels and running them. Also keep in mind that normally for proteins you have a vertical polyacrylamide gel. And if you see this acronym, SDS page, that is a protein gel technique, and this stands for sodium dodecyl sulfate. And this stands for polyacrylamide gel electrophoresis. Polyacrylamide gel electrophoresis. And you'll just see SDS page, all in caps like that, with a hyphen in between, a lot in papers. They're talking about this. Alright, so once you have separated your proteins, you've got them in order of size. They're kind of safe in this gel, but you usually can't see them. You've got a clear column of proteins. Whether you loaded total protein or partly purified protein extract, you can't see them. So normally the next step to be able to detect what you've got there is something called blotting, as in Western blot is proteins separated by size and captured on some type of membrane. And there are two big ways to do blotting. The purpose of it, you want to get the proteins out of the gel get them out of the gel and onto a solid, stable surface, which is normally called a membrane. So it looks like a little paper right here, a membrane, and they are usually shiny, and sometimes they are made out of nitrocellulose, and sometimes they're made out of other advanced materials. But you want to get your proteins onto a membrane. How do you do that? How do you get it out of the gel? There are two ways to get it out of the gel. This is the modern method. probably should only talk about the modern method. So in the modern method, you sort of sandwich your protein gel, which you have carefully taken out of the apparatus that you ran it in, and you have not dropped it on the floor and broken it. And if you did, you will put it back together and try to do this, and it'll work okay, but better if your whole gel is still intact. And you sandwich it between some specialized filter paper, the membrane that you want to get it on, more filter paper, and the modern method also uses current to pull the proteins out of the gel. Notice we've still got negative and positive poles, and you want to get the membrane on the positive side of the gel. The proteins are still negative, they still migrate to a positive, except now we have laid this gel down on this membrane, and the current is taking the ladder of proteins, like pretend that they're spread out like this, like these rows of buttons. Take the ladder of proteins, and they're still in their same positions, and now they're going to migrate down onto this special membrane paper, still by electrical field. This is called, well, maybe this is called blotting now. When I was in school, it was called electroblotting, because it's better than the old way. So then you get your proteins on the membrane. Usually you have to cross-link, fix them onto the membrane. Sometimes you do this by putting it under UV light, or you could use a certain chemical. That makes sure that the proteins stay on the membrane. The membrane itself also is positively charged to help them stay on there. And now they're captured onto a paper. And now they're kind of stable, generally stable. You've got separated proteins, separated by size. You've got them on a membrane, so now you have blotted them. And still, if you were to look at this membrane, it looks like that. You can't see anything. You got to have faith that you did all this well. And the next step is to make them become visible. Make sure I said everything about blotting. Yes. How are you going to make them become visible? Detection. How are you going to detect them? You've got them on the membrane, and your challenge is to make them show up. There are two major ways to make proteins show up. The first way is to bind antibodies that are specific to the protein. If you had immunology, or if you learned some about antibodies in another class, they are shape specific to the protein. Antibodies binding to the protein somewhat depend on the protein maybe slightly folding up again. So bind antibodies on and then make the antibodies visible. Or another way is that before you ever started this, you artificially tagged the protein with a molecule that itself can be bound to antibodies, and then generally how that would look. Let's start with number 1. Okay, you run the protein through the gel, you get it onto the membrane, and here is our membrane lying down. And your particular protein that you're interested in went to this place, and you can't see it yet. Went to that place, you can't see it yet, but it's there. It's there like in a ghostly outline. It's still got some shape remaining. And you could add onto this antibodies, which I'm drawing as the upside down Y, that are specific binding only for that shape. You could put them all over this. You actually end up soaking the paper in antibody solution. And the antibodies will bind to this. Remember, there's not just one of that protein there. There's maybe thousands that migrated to that spot and stopped. And you put the antibodies on, and they bind here, and they don't bind anywhere else. Only that place, and they're still invisible. But these antibodies often are enzyme linked. They are specially made by a company, and they're linked onto an enzyme covalently. And that enzyme can turn some type of substrate into a colored product. And we're going to use this type of system later in the semester when we do ELISA assays. It's the same type of thing. You've got an antibody that's got specific binding to only one thing down on this end. And that antibody is also linked to an enzyme that does a color conversion process. Takes a clear substrate and turns it into a colored product. So if you were to be using this method to analyze this blot, you would go through the following steps. Number 1, blocking. The membrane is smooth and specialized in advance, but it's still got crevices and cracks that antibodies could maybe bind to inappropriately. So normally you would put non-fat dry milk solution, which is really low tech. You would soak that membrane in a solution of non-fat dry milk, and the milk proteins are kind of like a blanket of snow, making everything look smooth and blocking up the holes in this membrane and preventing non-specific antibody binding. So this prevents non-specific antibody binding. Wash. I'm gonna put wash in between. There's a jillion wash steps. You wash, and then you put something called primary antibody, which is specific to the protein. So this one is protein specific. You wash. And then you put something called the secondary antibody. I have not drawn that over here. But this would be an example of primary antibody. Its arms specifically grab that protein and nothing else. Yes?
What do you wash with?
Wash buffer. It's not just water, but it's basically water that is salt and pH balanced to baby protein. Baby isn't verb, to caudal proteins. And it's just to remove extra milk, just to remove extra antibody. So normally you would have this little paper membrane in a dish, about like that. And if you're in a good lab, it's on a rocking platform or a rotating platform. It washes it for you, and you don't have to stand there and tip it back and forth. But that works too. And then you're just putting in 10 mils of block, pour it out. 10 mils of wash, pour it out. 10 mils of wash, pour it out. There's usually a lot of those. And you just keep adding these things, and we're going to do this actually later in the semester. Generally, same steps. That's a good question. The primary antibody specifically binds to this protein, and the secondary antibody is actually this, what I was drawing right here. This antibody, its binding sites specifically bind that part of the primary. So if an antibody is a Y, I'm an antibody, I'm a Y. And if I am the primary, my hands are grabbing the protein that I care about. My feet are bound by the secondary antibody. And so you make this tower of antibodies, which is kind of cool to imagine. Primary stands on the protein, the secondary stands on the primary with the enzyme linked on. There's washing in between. And then step 4 would be the enzyme substrate. So after you've got the secondary antibody on and you've got a tower that tall, all this is still bound to your membrane. The membrane's in the dish. You're making sure you keep it wet with various washes and so on. Then you add the substrate. And remember, this is stuck. It's stuck in this place, this band right here. It's stuck. It can't go anywhere. So the only place where you get a clear substance turning into a color product is where that band is. Right there at that spot. And that's how you make a certain protein band visible when there are actually 10,000 different kinds in a column running down through there. Now you can see your protein. Here's the crummy part. If you just discover a new protein, you don't have an antibody to it yet. It takes a while to get an antibody to that protein. That's when you usually use this method first. Do you know how you make antibodies, commercial antibodies? It's really horrible. I'll just describe it to you. You purify a lot of protein, you get it really pure in large amounts, and you inject it into a rabbit and you wait. And the rabbit senses it as a foreign substance, it activates the immune system, it makes antibodies against that protein, and its blood is full of them, and then you kill the rabbit and you get the antibodies out of its blood. I am serious. That is the way. So when you hear about commercial monoclonal antibodies, they grew in rabbits. It's really unfortunate. That's how you make them. I never had to do that for my protein. I used the tag approach. So I was working on a protein, I was interested in studying it, and to do this number 2 method, I'm going to erase now this. The steps at the end are kind of the same, generally the same. To do this number 2 method, does not require as much rabbit cruelty. That marker's not talking. The number 2 method, you genetically engineer your protein to have in-frame tag, an in-frame tag. And this is surprisingly easy to do. You engineer your protein to have an in-frame tag. So if this is my protein, one end of it has some particular extra amino acids of a known structure, and they make their own little like domain. And this is bound by an antibody that someone else has already made antibody to that tag. So examples of these tags can be named things like flag tag. It may not have a hyphen. It stands for something, FLAG. Flag tag, MIC tagged, HA, which stands for hemagglutinin, I think. HA tag. And the identity of these has nothing to do with your protein. You're just adding it to your protein so that you can grab it later with the antibody that someone else has gotten out of rabbits. And antibodies against these are cheap. Antibodies against these are cheap and really plentiful, and you don't have to purify tons of your protein and have somebody inject the rabbit for you and wait months to get an animal. Looking to see if I covered all the tags that we're going to see today. Those are not the only tags, but those are some really common ones that you might see in papers. CMYC, sometimes it's called CMYC, flag tag HA. So those methods combined will let you make your proteins visible on the blot. And then you've got bands of certain sizes. This part I'm going to skip over because it's not, I'm not sure how long things will take today. Is describing a protein I'm going to show here in a minute. The next thing actually I want to show is things like this. When you come to seminar papers and you see Western blots, you see this. They've done all those things that I just said to get to this point, but they don't really talk about them because it's not worth talking about because people in this world do it all the time. But students aren't used to it. And so when you have a Western blot in your paper, you're looking at this, and what this is a picture of a piece of a big membrane that was originally that big, and it's pointless to show all the blank parts of it because you have space constraints in the paper. So this is a cut and paste of a piece of membrane that I did a Western blot on, and the bands are showing up like that. So what I want to do in this next section is talk about, all right, when you see that, how do you interpret it? What does it mean? And there are a common set of abbreviations that are used on these, and what do those mean? And this will greatly help you with your seminar presentation, or when you see Western blots in the future, you'll know what they mean. This is going to be really useful. So even if you don't do this work, you're going to see these often in either your seminar or somebody else's. And in the past, I have been very not harsh on asking questions about Western blot because I know nobody knows how to interpret them, but those of you who are presenting next year, who would be presenting next fall, if you have a Western blot, I can ask really good questions about it. But don't let that deter you from picking a paper with a Western blot. Okay, so interpreting these. And you've actually got this on a slide, and also I've got some papers I'm going to give out to you because this is the figure, this is the legend that goes with it. I'm not showing the A and B part. But these are the methods behind getting to this. I'm not gonna so much go over the methods, but if you're gonna work in a lab, you might want to keep these and then compare these methods with what I've been talking about and see if you can follow what they did. It talks about how I made a flag HA tag, how I did the extraction, how I got the protein out, all the stuff I did. You'll notice in there I did a Bradford assay, and I don't describe it, but I cite it. So you've already done Bradford assay, which is a really standard technique that you would start with. I'll give you 2 minutes. I would fail paper passing. It shouldn't be this difficult. It's not too bad in here, but in principles, I have like 13 people in each row, and it's just ridiculous.
Okay, we're good?
I've got a copy of
I'm just giving to those two. Since you've got them both right. This figure 6 down here might become a lab exam figure. I think I'm not going to talk about it today, but see this figure 6 down here? I am likely to put that on the lab exam and ask you questions about it, and they will not be too difficult if you follow along with what I say next. These are the protocols to get to C. Those are optional, but very useful if you're going to do molecular lab work. Figure 6 down here at the bottom is going to be a lab exam figure, and I'll ask you questions about it. Okay, let's look at this picture up here. How many wells did I use? And how do you know? What do you think? 10, yeah. I've got 10 bands. I use at least 10 wells. A lot of researchers, as many wells as there are, you use them all so that you don't have to do as many gels, honestly. And what you're showing across the top is, what is being shown across the top is the genetic background, or what is different about what I'm looking at in each one of those. So what I'm looking at here are proteins that I extracted, and these are genetically engineered proteins. So when you see that they say AVRB HA, this is the protein that I'm actually interested in, and this is the tag. And so I extracted proteins from a bacterium that had a plasmid in it that was overexpressing that, an engineered protein, and that's what I loaded on the gel. So that was one, and that one is not showing up very much. It wasn't detected. This one, G46D HA, is a mutant version of AVRB. So this is a version of my protein where the original amino acid at position 46 was glycine, and then it changed to aspartic acid. So it's a mutant. That's the only thing wrong with that protein. It still has the tag. And then these others are other mutants. Okay, out to this side, this is a size marker. That's a size marker band. So to put that band on there, I had to, at some point, run a size standard along with my protein and figure out that 37 kilodaltons is just slightly bigger than this. Are these proteins bigger or smaller than 37 kilodaltons? They're smaller. Yeah, you can assume that the original orientation of this membrane paper was with the wells at the top. And if 37 is on that side, these are smaller. I think this is a 34 kilodalton protein. So they're smaller than 37 kilodaltons. And in this picture, I'm only showing like one row of proteins. I'm not really that interested in how big they are. In this, I was not looking at how big they are. I want to know how stable they are. So hopefully, yeah, down here, my figure legend says something does not result from insufficient protein accumulation. So I wanted to know, does being a protein mutant cause the protein to be less stable, or is it just less functional? So this is saying that, well, all of these actually accumulate at least as well as the wild type. So it's not that they're getting degraded and losing function, they're just not functional. They're there, but they're not functional. This, what that says is anti-HA. That's the primary antibody that I used on this blot. That's the antibody that I added in after the milk block step. And it's going and binding to wherever proteins have a HA tag on it. And if you notice down here, it says down here in the legend, a 1 to 1,000 dilution of anti-HA HRP. Oops, HRP. This is the enzyme, and it stands for horseradish peroxidase. Horseradish peroxidase, and we're actually going to use an antibody in this semester linked to horseradish peroxidase. I forget what the substrate is now, but we're going to use it in like a month. And so any place where that bound, it bound to the HA tag, and then I added the color substrate, and then bands showed up, and it's a happy day. So that's a really simple look at a very simple piece of a Western blot. I'm just looking more or less. I'm not looking so much at the size changes. They're about the same size. They are denatured, so it could be that, you know, some of these have a little bit bigger weight of amino acids than others, and they slightly shift, but I'm just looking at more or less. Is it there, is it not, and so on. Okay, here's another blot that I did that is more complicated. And this one, I seem to see more students looking at blots like this in their papers, where you've got multiple, you've got a lot more lanes, and there's pluses and minuses all over the place, and there's size differences and so on. So let me go through this one. Okay, let's start here on this side. You could count the lanes. This is a longer lane. 14, so this was a 15 lane gel. I had cut off the size marker over here. And what this stuff is showing is what combination of these three things were in the sample that made up this lane. So let's look at this one first. This, no. That's a whole different kind of protein than what I was talking about here. But it's a mutant version of this protein. Both of them have HA tags on. So this is a wild type protein, this is a mutant protein, and they both have HA tags on them. And in this lane, minus minus, those weren't expressed. I never put these two into I think it was a leaf that became the source of this sample. But it did have this, which is Arabidopsis thaliana Rin4 protein with a MIC tag. These are artificially tagged. These have a different tag than these. So what is in this lane is only that. This lane has this. This is in every place where you see this line going across. So this lane has this and this, but not this. This shorthand system is used a lot for western blots because it saves space. And in this particular lane, I was expressing this and this, but not that one. So they never were all three together. So that system continues all along through here. This is a Arabidopsis protein. These are soybean versions of that protein, and this one is with no RIN4 at all. So that tells you what was in that lane. So I did that blot, ran it, separated it, and the first time around, I'm using antibody against a MIC tag. I'm using a MIC tag antibody, and that will show me what's going on with those. My antibody is only against the MIC tag, it's not against these, so I can't see these in this section. This is showing me where the MIC tag proteins are. Okay, knowing that, which is the biggest protein out of that group of RIN4s, which is the biggest protein? There's actually two that could be it. 4A and 4B are slightly bigger than 4C, and all of them are bigger than the Arabidopsis version. So you can tell a lot from these. Why don't I see any RIN4s? Why don't I see any bands over here? I didn't have any particular samples that had a MIC tag that could show up. So this is like a negative control. What other questions could I ask about this?
I have a question.
Yeah.
Why is it that A and B are bigger than, why do you say A and B is bigger than C?
Why do I say that they're bigger?
Mm-hmm.
So this is a piece of an original membrane that was bigger. And they always show it to where this piece, the wells were up here, and the proteins were running down to the other end. And if shorter ones run faster, shorter ones end up lower down. That's how I know. So when you see bands like this, the ones that are lower are smaller than the ones that are bigger.
Okay,
That's
okay.
why. And that's a good question.
Because I was thinking size-wise of the dots. And
Oh, the blobness?
yeah, that's what I was thinking.
What would be a better question for me to ask was, which one has a bigger molecular weight? But what does the blobness tell you? How much? Yeah. So this band is a higher molecular weight than this band, but there's a lot less of it. There's a lot less of it. And there's a lot less of this than this. Which means that if I have this by itself and then I add in that mutant, something about, or let's compare a different way. Okay. In this lane, I only have soybean RIN4B. In this lane, I have added in a wild type bacterial protein, and suddenly soybean RIN4B is gone. It's not there anymore. Actually, because this bacterial protein destroyed it somehow. It's gone. The mutant bacterial protein does not destroy it as well. That's a lot to tell from this. But that's what these let you tell if you set them up the right way. here's some other things you can tell from this. This mutant doesn't destroy this one very well. It doesn't destroy it at all, basically, because these are the same, about the same. This mutant destroys this one, kind of. It has no effect on this one. Kind of cool. That's what we can tell from this. So then, okay, that's what I really wanted to know. This is telling me something different. This is telling me, do these proteins not work well because they're not processing? They're supposed to be processing. So here's the wild type version. If there's no RIN4, let's compare over here. The wild type version is supposed to be this size. Wild type AVR RPT2, when you look at it with HA antibody, so these are seeing HA, these are letting me see the bacterial proteins. It's supposed to be this size. And I put a helpful arrow for people to say, this is the process size, this is how it's supposed to act. And notice that the mutants don't get to the small size, they say the big size. Mutant big, wild type small, mutant big, wild type small, like so, like so. So the mutants probably don't do their work on the soybean protein very well because they didn't process correctly. And I know that by the sizes. Here's another important point to add. Between here, so this is step 1, I looked at what RIN4s were doing by looking at MIC tag that were on those proteins. In step 2, I'm using the same membrane, but I took all of that off. It's called stripping. I stripped it. I'm not making up that word, that's the real term. I stripped that membrane and took all the antibodies and color products off, and I reused it with a new antibody. That's kind of why these don't look as nice as this, because this is like, when you erase your whole paper and you use it again, it doesn't look as pretty. But you can only really look at one antibody at a time. Well.
What do you strip a membrane with?
I'm trying to, you basically use a pretty harsh chemical that disrupts the antibody stack binding to the protein, but it doesn't take the protein off the membrane. But I don't remember now what it was. At that time, I never really thought about the chemicals. Just use them. Okay, we are getting close to the end. Another thing that you will frequently see on Western lots is, well, I'll get to that. Okay, this is from another group that continued some of this work after I quit and had kids and became a teacher. They're looking at a specific antibody to RIN4 itself. So compare back here, I was tagging RIN4 with a genetically engineered tag because we didn't have RIN4 antibodies. We didn't have RIN4 antibodies. Somebody now has gone to the trouble to hurt the bunnies, make actual RIN4 antibodies specific to RIN4. And so they are looking here, you can see what's in each lane, and they're looking at RIN4 accumulation. And I didn't actually read this paper, but this looks like one of those graphs where they're just checking that, is there enough and is it the right amount? Yes. This is a null mutant, I think. It doesn't make RIN4 at all. But then there's this. And sometimes if you see a lower down section on a Western blot And it's bands that are the same size and about the accumulation. A lot of times this is a loading control depiction. And this actually doesn't really have anything to do with their experiment, but what it's showing is that they loaded equivalent amounts of total protein. Like way back in the beginning, when they loaded their gel, let me escape and go back. Let's go back to our gel rig picture. Way back here, when they were loading the gel, were they loading the same amounts of total protein back here? You need to know that before you can say anything about protein accumulation. Because if you messed up back here and your thumb pushed too hard and it spurted out everywhere, and then later on you saw, oh, my protein is disappearing. No, it's not. You didn't load it right. So a lot of times in Western blots you'll see these lower down extra sections, and even students that like understand Western blots, they'll come and ask me, I don't understand what this is. And it's because it has nothing to do with the actual experiment. If you're just checking something, did you load correctly? And there are different ways to do loading controls. CBB, or if you see Kumasi, they're calling this down here, stained with Kumasi, CBB. In that case, they took the gel after they ran it, and before they blotted it, they poured Kumasi blue, which is part of the same dye in the Bradford reagent. And there are certain proteins within cells that are so prevalent that they will stain in big bands, even just in the gel, and you can see them. Sometimes it'll be Rubisco. This one, they're not saying what Kumasi stained band this is, but it might be Rubisco, which is the most common plant enzyme and it's present in huge quantities. So sometimes the loading control will be Rubisco. Sometimes in animal systems you see actin. Like you'll see a row of big, fat, nice, even black bands down here, and it says anti-actin, and you might be like, I don't think they're doing anything with it. They're not doing anything with actin. There's just lots of actin in the cell, and you can use it as a check that you've loaded equal amounts. Actin is the one for animal systems. And one of these, yeah, there's Rubisco on your lab exam figure. Mine doesn't have one. What did I do? I did do something. So on mine, samples were normalized to equal protein concentrations. So I did Bradford assays on my samples before I loaded them into the wells, and I'm kind of saying, trust me, I loaded equal amounts. You got by reviewers. That's what you need. But really good Western blots would have a loading control. This one, I would have to go back and look at the figure legend about what I did. It's probably the same thing I did before. This one has Kumasi steam band. You can see on this one they've got Rubisco bands. That's how you know you loaded the same amount. Okay. I think that's good. Let me ask a few questions to you about, let's say this one, as a practice, to sort of give you an idea on the lab exam what I might ask. I might ask you, for example, like put an arrow in this lane. It's easier to put it down from the top. And ask you what proteins did I expect were in that original sample when I loaded it? Or even how many was I expecting? What's in there?
There's 13 on
Yeah, I would think that what I had loaded in there was this. So this line means that all three of these have this. So I expect that I have soybean Rin4B. And I expect that I have this particular mutant of the bacterial protein, but this is not there. On an exam, I would take these off. And I might ask you, what does this actually represent? What does this actually represent? Even if I took off the labels. So you would have this label still there, this label still there, and you have this information. Because I've noticed a lot of journals now, they don't put these helpful. They just expect you to know. So that type of question. I might ask you, which is bigger, full-length ABR RPT2 or processed, and how do you know? You do that by seeing which one migrates lower down. Other possible questions as well. Do you understand it some? I know you understand it some better than you did before.